Hi, everyone. For those that have already joined, we're just going to wait a couple more minutes before we get started, probably right around 11:05. Thanks. Okay, great. So I think we're going to get started. Hello, everyone. My name is Jonathan Nassi, and I'm the Director of Translational Science here at Inscopix. On behalf of the science team and the entire company, I'd like to welcome you all to our Inscopix Journal Club. We've begun a new series we'd like to share with the Inscopix community in an effort to help keep the scientific spirit strong during these difficult times. We'll be periodically discussing publications of importance to the field of circuit neuroscience, often featuring Inscopix and Vista imaging or Invoke combined imaging and optogenetics, and we hope that you can join these discussions from your virtual offices at home. You can send us questions via the chat window throughout the presentation and for up to 5 minutes after the presentation concludes. We'll do our best to get to most or all of the questions, but we will have a hard stop right at 12 p.m. Pacific time. So today, our senior field scientific consultant, Vardhan Dani, will be presenting a recent Neuron publication from the Tonegawa lab at the MIT Pekauer Institute. We're really excited to discuss this paper with all of you, so thanks very much for joining. And now I'll let Vardhan take it from here.
Hello, hi everyone. Jojo, can you confirm that you can hear me? Okay, cool. So hi, welcome to our journal club. I wish we met in better circumstances, but let's try to make the best of what we have. So as Jojo mentioned, hang on, let me put this in presentation mode. I will be discussing this paper that recently was published in Neuron and came out of Susumu Tonegawa's lab at MIT, and exploring the role of specific cell types in the basolateral amygdala in fear extinction memory. So just to take a step back and broadly speak about classical conditioning paradigms, you can always train, or animals can learn to associate positive or rewarding stimuli like food with seemingly neutral stimulus like a bell ringing. And if you repeat it a few times, then the animal can respond as though it's responding to the unconditioned stimulus just by the ringing of the bell. And the same applies in fear learning paradigms, which is often used in experiments to as a model for classical conditioning, where an animal can learn to associate a neutral tone with a somewhat mildly noxious stimulus like a foot shock, and then presentation of the tone itself can elicit a response as though it was for the foot shock, which is usually quantified in terms of the amount of freezing. And it's also important to remember that while animals learn this very quickly, they can also learn to not remember this, which is generally in the context of fear conditioning referred to as fear extinction, which is the topic of this paper. So as I mentioned, fear learning is an important mode for associative or learning or classical conditioning, and it's an experimental model for studying neural circuit mechanisms underlying classical conditioning. And you can imagine that this is somewhat relevant to pathophysiological states taken to the extreme. It can be relevant to states of anxiety or even PTSD, where people might have long-lasting memories of unpleasant events. And so it's not only important to study how the neural mechanisms of how one might learn fear-related memories, but also how one might overcome those memories through extinction mechanisms. And very broadly speaking, some of the neural or circuits or the brain regions that are thought to be involved and are the circuits that are thought to underlie storage or learning and storage of fear memories are shown on this panel on the right. It's mainly a lot of labs have studied the circuits in the medial prefrontal cortex, hippocampus, and amygdala. But today's discussion will be focusing on circuits in the amygdala. So one model that has been championed a lot over the last several years by the Tonegawa lab for the cellular mechanisms or circuit level mechanisms for learning and storage of fear memories is the so-called engram cells. Engram cells are shown experimentally in sort of this cartoon, and I would highly recommend reading one of these two reviews or both of these reviews if you want to understand a little bit more about them. But very briefly, if you were to activate a subset of cells using somewhat noxious stimuli like a foot shock to the animal, and then have a genetic way of labeling only in an activity-dependent manner only cells activated during that foot shock, such that they can be expressing, let's say, an optogenetic effector. And then if you activate that optogenetic effector, you can induce the same fear-like or freezing response in the animal without the need of the associating context, so on and so forth. So the idea is that the association of the fear or the fear memory itself is stored in subsets of cells, which are then reactivated when that memory is recalled. So along those lines, in this paper that which we're going to discuss today, the authors wanted to look at the putative role of, or are there engram cells associated with the extinction of fear memory? And if so, what do they look like? So just another piece of background for which is relevant to this paper. In previous studies from the Tonegawa lab, they had shown that there are most of the principal neurons in the amygdala can be subdivided into two anatomically and functionally distinct cell types, which are labeled by these two markers called RSPO2, which labels mostly the anterior portion of the BLA. These are sagittal sections as shown as examples, where RSPO2 is labeled in green. And these are in situ hybridization experiments, fluorescent in situ hybridization or FISH experiments, and triple P1R1B, which is related to a protein phosphatase. I don't really know exactly what these molecules do, but that's what they stand for. It's a protein phosphatase regulatory subunit. And this marker seems to be expressed mostly in the posterior BLE. And then another study, they had shown that these not only is there an anatomical segregation, but these are functionally different in the sense that the RSPO2 neurons seem to be activated for positive or rewarding stimuli, whereas the PPR1B, sorry, it's the other way around. I mix that up in my head. The RSPO2 neurons reward more to negative or aversive stimuli, whereas the triple P1R1B respond to positive or rewarding stimuli, such as food-seeking or appetitive stimuli. So going back to the paper, the authors started off with a typical contextual fear learning paradigm, and shown here, this is basically figure 1 in the paper. Where they to study fear extinction, they expose the animal after a habituation period. They expose the animal to a behavioral chamber which sets the context, and they learn the animal learns to associate that with a foot shock, followed by day 2, where it learns to actually basically unlearn the association, where it goes back to the same chamber in the same context, but now there is no foot shock. Although initially the animal would show some freezing because it's learned to associate that same chamber to a foot shock, but over time it actually unlearns it because there is no more foot shock. And then on day 3, they test for this extinction retrieval memory, so to speak, where it's again exposed to the same chamber, and they assay the amount of freezing. So as you can see over here, during the training on day 1, as the animal experiences foot shock, it increases the freezing, following the blue graph here. And as it on day 3, when the animal has learned to forget that association, the freezing reduces. Now the control groups over here are a non-extinction group, where instead of returning to the behavior chamber where it learned the association, the animal is returned to the home cage and then tested for extinction learning. And another control group is a non-shock, where the animal does not experience any shock at all. And as you can see, the animal, when it does not return for extinction training, it maintains a higher amount of freezing. So it did not learn to dissociate the fear memory. So basically, there was no fear extinction. So, the authors first look for these two markers that I just talked about a couple of minutes ago, the RSPO2, and try to co-label it with immediate early genes and see whether these neurons are involved in the fear learning versus fear extinction. And so shown here is the co-labeling of immediate early neuron, immediate early marker CFOS in the RSPO2 neurons. And they see that they are highly expressed only in the non-extinction group, where the animal did not get a chance to unlearn the fear memory. And on the bottom panel are basically example of the fluorescent in situ hybridization data. When they look at the other marker, the triple P1R1B, they see that the CFOS expression seems to be higher, more towards the posterior BLA, where that marker is enriched, and it seems to be higher in the animals that actually experienced the extinction of, or was learned to forget the fear memory compared to the control groups. So to then test whether this actually happens functionally in vivo while the animal is behaving, they use Inscopix's Envista miniscopes, and their experimental paradigm is shown on this slide, where they use Cree lines specific for either the RSPO2 or the PPP1R1B to express GCAM6 as the calcium indicator, which will be the reporter for neuronal activity in a Cree-dependent manner in one of these two cell types. And they record neuronal activity at different phases during the training and testing of the extinction retrieval. And that's sort of summarized in panel D over here. They record activity during an habituation period, then during the fear learning itself, which is shown here in the shock, and then during the extinction training, where initially actually the animal remembers the fear memory, so they treat the initial period as a fear retrieval period, followed by an extended period of extinction training, where the animal is just remembering to forget the association because it's in the same context and not experiencing a shock. And then they come back on the third day and test whether the animal can actually retrieve the extinction memory. And these figures over here in B and C just show the placement of the grin lens implants in either the anterior BLA or the posterior BLA, remembering that RSPO2 is mostly enriched in anterior BLA and the PPP1R1B is mostly enriched in the posterior BLA. And that's how they separate these two populations in addition to the Cree lines themselves. So when they look at the activity of these neurons, they find that, first of all, if you look at these pie charts, they find that most neurons are not that sensitive to changes in activity during the behavior. But among the neurons that are sensitive, they find that RSPO2 neurons are mostly activated, which they call as sort of upregulation, labeled in this orange sort of piece of the pie. And the blue indicates neurons that are downregulated. So RSPO2 neurons, amongst the neurons that do show changes in activity, most of them seem to be activated during the shock and fear retrieval, but not so much during extinction training and extinction retrieval. Whereas the other subpopulation of neurons, the PPP1R1B cells, are amongst the neurons that do show changes. Most of them are not activated during shock and fear retrieval, but they seem to be more activated during extinction training and extinction retrieval. And this is also sort of graphically represented in these heat maps showing the delta F/F changes and these example traces of individual neurons, where they're comparing for RSPO2 cells the period between habituation and shock, and they show upregulation, and here's an example of the downregulated RSPO2 cells. And on panel I and J, they show neurons that are showing not a big change in activity in between habituation and shock, but actually are upregulated during the extinction retrieval. So just want to quickly check, are there any questions, Jojo, so far?
No, Vardhan, I think you can keep going along, but I will mention that by all means, if people do have questions, please feel free to do so via the chat, and we can occasionally interject those. Otherwise, we'll field those after Vardhan has finished his presentation.
Okay, so I'll keep marching along. So they wanted to now test. They've seen that that the RSPO2 population seems to be activated during shock and fear retrieval, whereas the PPP1RB are activated during extinction training and extinction retrieval. Is this really functionally relevant? They've seen that there is a correlation in the activity of these neurons, and it is consistent with previous data that RSPO2 neurons respond to more negative or noxious stimuli, whereas the PPP1R1B neurons respond more to rewarding or appetitive stimuli. So see if this is functionally relevant. What they do is they express either channel rhodopsin or archaeorhodopsin, which is the inhibitory opsin, in either the RSPO2 Cree or the PP or PPP1R1B Cree. And the animals, the control animals are Cree negative in either of these conditions, and the animals undergo contextual fear conditioning, followed by the same paradigm, essentially the extinction training and extinction retrieval, but they are going to be, their activity of one of these cell types will be manipulated during the extinction training, which is also overlaps with the fear retrieval actually. So what they see is that animals undergo normal fear learning, which is shown on this graph on panel D on the left-hand side and on panel F on the left-hand side. And in the case of RSPO2 animals, when they are inhibited, or if the PPP1R1B animals, when they are, or the neurons, when they are excited, the fear extinction memory seems to, or retrieval seems to accelerate. So just as is shown over here, a little bit in a mild in the RSPO2 Cree animals, but pretty prominent acceleration of the extinction memory in the PPP1R1B animals. And conversely, if you excite the RSPO2 Cree neurons with channel rhodopsin, that actually prevents the expression of fear extinction, which is indicated by higher levels of freezing on day 2 and day 3. And if you inhibit the PPP1R1B neurons, that also leads to blocking of the extinction learning. So again, suggesting that the PPP1R1B neurons are putative, quote unquote, engram cells where extinction learning memories are getting stored. So actually, this is just showing that instead of manipulating the PPP1RB neurons in the extinction training itself, they are now manipulating the activity of these neurons during extinction retrieval on the final day when they're testing extinction retrieval. And when they excite these neurons, they don't see a lot of effect in the freezing, probably because it's already reached a floor in terms of learning. And but when they inhibit these neurons, they do see an increase in the amount of freezing, suggesting that it is now blocking the retrieval of the extinction memory. Again, supporting that the PPP1R1B neurons are important in learning as well as retrieval of the fear extinction memory. So, hold on a second. So they continue on trying to test whether these are really engram neurons using the paradigm that the lab has used previously to show that that neurons are actually subsets of neurons are actually engram cells, and they can be reactivated to recalling the same subset of neurons would reactivate the memory. So the experimental paradigm they use is described here. They essentially express 3 sets of AAV viruses. One that drives a tetracycline sensitive transactivator in an immediate early through an immediate early promoter of CFOS, as well as it is Cre dependent. So that way it is restricted only to a subpopulation of the P1 R1b neurons. And then they express either a channelrhodopsin EYFP construct or EYFP alone as a control to manipulate these neurons and see whether activating these neurons can actually elicit the fear memory again. And so the way this works is that they first treat either the EYFP or channelrhodopsin expressing mice to contextual fear conditioning. They go through the extinction paradigm, and all this time the animals are on a doxycycline on diet, and they remove the doxycycline only during the extinction retrieval period, so that they can label only the neurons that are activated during the extinction retrieval phase. And then the animal returns to the home cage, undergoes another set of fear conditioning, but this time instead of having the animal recall the memory based on what it's learned during the contextual fear conditioning, they only provided optogenetic excitation and see whether that elicits the same memory. Oops, sorry. They also introduce another control group where, in addition to just the EYFP alone construct, they also have these channelrhodopsin expressing mice where they try to express the channelrhodopsin only in an activity-dependent manner, only when the animal has undergone this training but is now returned to its home cage just during this home cage period. So now only the neurons that are not related to the supposedly not related to the extinction retrieval will get labeled. So when they test these neurons in this paradigm, when they test these animals in this paradigm, they find that the animal undergoes freezing during the learning phase, undergoes normal extinction, as seen by reduction in freezing during the extinction retrieval. And then later on, when they test on day 6, over here, where they have now labeled the neurons that were activated during the extinction retrieval, they see that when activating those neurons actually enhances or accelerates the fear extinction, suggesting that again these neurons are functionally important in driving the extinction retrieval process. And that's just summarized here in these bar graphs. So in the next set of experiments, they try to so they try to do the same thing in the RSPO2, which are the neurons that are more relevant to fear learning rather than extinction. And they see that they don't actually see a lot of change in when they excite these neurons, but when they inhibit the RSPO2 neurons, they actually see an increased amount of freezing when they try to label these neurons during fear retrieval. So this actually suggests that these RSPO2 neurons are probably and the PPR1B neurons are mutually inhibiting each other. And this was shown in a previous study through electrophysiology experiments. So this is kind of consistent with their previous data. So I just want to quickly check in. Jojo, any questions?
Nothing. I mean, there's a couple questions here, but I think we can probably likely field those and discuss once you finish up. Nothing too critical to cover at the moment.
Yeah. Okay. So in the next set of experiments, as I'd mentioned earlier, they had also shown that the one subset of these neurons, the triple P1R1B neurons, are also activated during somewhat rewarding stimuli or appetitive stimuli. So they try to label that subpopulation and see whether these are overlapping whether there's an overlap between neurons that are labeled by activation during say appetitive stimuli like water or food presentation versus neurons that are activated during fear extinction. So this is a bit of a complicated setup, or so they basically all animals in this experiment undergo first a period of water deprivation, and then they are given water. So essentially, they are trying to label TTA and immediate early gene-driven TTA and EYFP. TTA dependent EYFP. They're trying to label neurons which are activated when the animals are presented with water after a period of water deprivation. And then these animals are separated into 4 groups, where the first group, the WW, undergoes further water deprivation, and then is presented with water. And after that, they euthanize the animals and look for immediate early gene expression. In the second group, the animal actually undergoes food deprivation and is then presented with food, and again is look for acid for immediate early gene expression. In the third group, the animal undergoes contextual fear conditioning and extinction, and then extinction retrieval and is assayed for immediate early gene expression. And the fourth group undergoes fear conditioning but does not undergo extinction and is tested for fear retrieval. So, sorry, this might be a little confusing, but this is how they try to compare whether there is an overlap between neurons that are activated with water and which is the only on day 2 when they present water to all of the animals which were water deprived. That's when you would expect these neurons to get labeled. And then on day 5, when you present them with either of these behavioral stimuli or paradigms, you would expect that the immediate early gene expression would reflect neurons activated during this last presentation of behavior or food or water stimulus. So when they look at this, they see that the overlap between CFOS and the EYFP in the posterior BLA, and so I forgot to mention that this is all being done in the posterior BLA, where the triple P1R1B neurons are enriched. They see that there is significant overlap in the first 3 groups, meaning the animals that most recently were presented with water, food, or extinction retrieval. But there is not a there's not a large overlap between the EYFP labeled and the immediate early gene expression in the animals that did not undergo fear extinction learning. So again, this is somewhat reinforcing the overall conclusion and also their previous findings that neurons in the posterior BLA are enriched are mostly these triple P1R1B neurons, and that they are mostly activated by either appetitive or stimuli or during extinction retrieval, but not so much during fear retrieval. So they again try to look at this using calcium imaging, using the Inscopix and Vista miniscopes. And they're trying to look over here at essentially subpopulations of neurons that are not only activated during either food or water presentation or extinction learning, but also how many of those neurons that are activated during, say, either food presentation or water presentation are also activated during extinction training and extinction retrieval, or fear extinction training or extinction retrieval. Trying to see whether these neurons are functionally overlapping. And so for this, basically what they're doing is in the experiment, they're imaging on day one, the animal is undergoing water deprivation. On day two, it gets a water reward. On day four, it's deprived of food. And on day five, it gets a food reward. So these will hopefully show neurons that are more activated or modulated by either water or by food. And on day six, it undergoes the fear learning paradigm and habituation, then shock, and then extinction training and extinction retrieval. But important to note here is that they are really looking to track the activity of the same neurons over multiple days. And as they'd seen earlier, only a small population of these neurons are activated by either of these manipulations. And some of them are upregulated and some of them are downregulated. And what they see, as they had seen before, is are consistent with their histology and immediate early gene expression, that amongst the neurons that are modulated in their calcium activity, a majority of the triple P1R1B neurons are upregulated in activity in response to water presentation, in response to food presentation, not so much during fear learning, which is represented by during the shock period, but they are more activated during extinction retrieval. And they try to look at the extent of overlap between water activated neurons versus extinction activated neurons. They seem like they show that there is a pretty strong overlap between these neurons, but not so much in the neurons that were activated by shock as well as water. The same with neurons that are activated by food and were activated during extinction retrieval, but not so much during food as well as the fear learning shock. And this Venn diagram basically shows the overlap of all the neurons that were activated during each of these phases. Again, it's important to note that these neurons the ability to extract this data was dependent on their ability to track the activity of individual neurons through each individual training session and over multiple days. And this graph just shows the activity of the neurons that were basically activated in all three conditions, with water presentation, food presentation, as well as extinction retrieval. Shown here is just a heat map of all the neurons that were showed activation in all three conditions. And the calcium traces are a representation of 6 of these cells in the heat map. And you can see that pre-water, the neurons are less activated, but they are more activated post-water. Same with food and same during extinction retrieval. Again, suggesting that there is a subpopulation of, even within this cell type, which is activated more during appetitive stimuli as well as during extinction retrieval, and there is a strong overlap between within that subpopulation. So then to test whether this subpopulation can also be behaviorally relevant, meaning can it drive the behavior, they again turn back to optogenetic manipulations where they label in the posterior BLA, which is again enriched with triple P1R1B neurons. They label these neurons with in an activity-dependent manner with either channel rhodopsin or just EYFP as a control. And in this case, the animals first undergo, in this experiment, the animals first undergo contextual fear conditioning, followed by extinction and extinction retrieval. And that's when this set of animals gets labeled with either channel rhodopsin or EYFP. And then they test whether they are able to drive the extinction behavior or manipulate the extinction behavior during optogenetic stimulation. In control group, or not really control, but a kind of control group is instead of extinction retrieval, this set of animals undergo water deprivation and then presentation of water, and then undergo either optogenetic self-stimulation or an optogenetic place preference test. That's the behavior that they're trying to test, whether these subset of neurons can manipulate, are enough to manipulate modulate the behavior of the animal. So the two behaviors that they are doing over here, as I mentioned, the opto self-stimulation is essentially the animal is given a choice between two nose ports, where it is given a small water reward at the beginning of the trial, and following that, the optogenetic stimulus is turned on for a certain period of time, and then it's tested over. I forget exactly how much it was, probably it was 40 or 45 minutes, but it is tested whether it has a preference between the port that was first presented with the water stimulus versus the port that was did not have any stimulus at all. So that's the port that had water stimulus called the active port, and the port that was did not have any stimulus was the inactive port. And if the animal has a preference with the active port, because it gets stimulated in that when it pokes its nose in that port, then it would show an increased incidence of increased amount of pokes in the active port. And they see this in the neurons labeled during extinction learning with channel rhodopsin, but not in the YFP neurons. Obviously, that's the control. And as well as the neurons that are labeled with water presentation, but not the ones that are that did not undergo an extinction training. The second behavioral paradigm that they used was an opto place preference, meaning a chamber where the animal is given a choice to be in one half versus the other. And if it goes in one half, only then it undergoes optogenetic stimulation. And again, it shows over here that neurons that are labeled with extinction retrieval seem to have a preference for on the side of the chamber where it receives optical stimulation, and it's the same as the case for neurons that are labeled during water presentation, but not in the control groups. So suggesting that neurons that are labeled either with extinction retrieval or water reward, they seem to be functionally interchangeable. And that's the main point that the authors are trying to drive here, that there is a subpopulation of neurons in the BLA, a subpopulation within the triple P1R1B neurons that is labeled, so to speak, as engram cells, and they are labeled. They function as engram cells either during rewarding appetitive stimuli or during fear extinction learning and these engram cells are sufficient to drive the relevant behaviors, either reward related or fear extinction related behaviors. And this is just a control for the other set of mice. Sorry, I just skipped that figure. But so to summarize, the authors show that the fear extinction learning involves formation of new engram cells within the BLA. These cells are a subpopulation of a cell type labeled by these P1R1B marker, which is mostly found in the posterior BLA. The P1R1B engram cells are functionally overlapping between appetitive and reward-related behaviors and fear extinction memory recall. So the broader point the authors are trying to make is that fear extinction memory, which basically involves omission of an expected aversive stimulation, is analogous or similar to a reward seeking or reward learning behavior. And there is now and the engram cells are probably the neurobiological substrate for both of these behaviors. So yeah, that's the paper. Please feel free to ask questions. And if you are interested in more publications using the Inscopix miniscopes, follow this link. Thank you for stopping by.
Great, Vardhan. Thanks for taking us through that paper. And those that are listening in, if you do have questions, we can stay online here for basically until 12 p.m. Pacific time. So we have about 12 minutes or so. There's already a few questions, and feel free to keep submitting them if you have those questions. Yeah, Vardhan, this is a really nice, I think this paper is a really nice demonstration of kind of this connection in the amygdala in terms of encoding of reward and fear learning. I think it'll be interesting kind of going forward to kind of probe how this relates to other brain regions in the reward processing network.
Yeah, I agree.
Dopamine signaling and all sorts of things like that. I think I imagine that the authors and probably others are interested in taking this kind of research down that path.
Yeah.
Yeah, a couple questions that have come in. Let's see. One question that came in that I also had a bit of a question about. I don't know how much you really know about these two different subpopulations of principal neurons in the amygdala to look at, but one question that came in was just asking to what degree these two populations are completely distinct. In other words, is there, are there cells that actually express, both of these genes, or are they, really mutually exclusive?
So I think to the best of my knowledge, and I'm basing this mostly on the Kim et al. Paper from which I showed some data, the first one over here, is also from the Tonegawa lab. I think the most of the non-overlapping populations are sort of well segregated among the anterior and posterior axis, as you can seen on this graph over here. So more towards the anterior are neurons that are RSPO2, mostly RSPO2 positive, and on the posterior side of the BLA, they are mostly P1R1B positive. There might be some neurons that are overlapping in between, but if I remember this paper correctly, those are not very large in number.
Yeah. Okay. And do you, I mean, in your background research, did you by chance come across any other differences between these populations? Like, do they have they looked at different kind of projection patterns, or?
I believe the Kim et al. Paper has some whole cell patch clamp data. I can't remember the conclusions of it, but they have tried to characterize these neurons in terms of their intrinsic properties, etc. What they also did show in that paper is that they seem to be mutually inhibiting each other, which sort of goes with their functional role, where one of them is activated during fear conditioning, while the other is not, and the other population are activated during extinction learning, while the first population is not. So there is the mutual inhibition seems to be consistent with that.
Interesting. Yeah, this mutual inhibition, this kind of interaction between the two populations is interesting to me.
Yeah.
And I was thinking actually using the Invoke miniscope, one could presumably image from one of these two populations and then optogenetically manipulate the other.
Possibly.
And it might be interesting to look at that.
Yeah, definitely. Yeah. No, that's a
great. There is overlap. There is some over. Like I'm just looking at your the AP kind of gradients, and it seems like you could probably sit yourself there in the middle and be in an interesting spot maybe to look at the
although one thing I should mention, and again, I'm not 100% sure, but if my memory serves me well, the inhibition is probably disynaptic. I mean, there are some interneurons in between.
But still, it'd be interesting to, even if it's
I agree.
A disynaptic kind of functional effect, it'd be interesting to look at that directly. Great. And then let's see, another question, if you can actually go to figure 2.
Sure.
Looks like there was a question about how they quantified the shock responsive cells. The comment said that it wasn't clear to them, just looking at the examples, the degree to which they really looked shock responsive. And so they're kind of wondering how that quantification happened. I assume they're referring to 2G and 2H maybe, right there. And I mean, I know, I mean, I was actually just looking back at just kind of the methods. I mean, they mentioned that, they compared the mean delta F over F of a 3-minute period after delivery of foot shock with that of a 3-minute habituation stage beforehand.
Exactly. So they're comparing it to the delta F by F in the habituation period versus the delta F by F levels during the shock period, and similarly for retrieval or training, so on and so forth.
And I would say in G, where I think we're probably looking at maybe average response, time locked to the shock.
To the shock.
It seems like there's clearly some cells that are
some upregulation. I mean, it's also important to note that this is not representing all the image cells. This is this graph shows only the ones that were showed some change in activities, and the main point was that there are, at least for the shock condition, is that most of them seem to be of the RSPO2 neurons are activated. The ones that did show some change in activity with respect to habituation, most of the RSPO2 cells showed activation, and a minority showed a drop in activity. Whereas if you look at the other subpopulation, again, really small portion of them are show any change in activity, but amongst those that do show change in activity, most of them are inhibited during the shock. Sort of highlighting this sort of dual, sort of functionally complementary role that these two neurons are seem to be playing.
Right. Excellent. Yeah, and then just one, it's not really a question, I guess a comment, just someone sent in a comment saying that they were pretty impressed with how the GCamp imaging looked in figure 2. I guess, I don't know if you showed 2B and C, but where they showed the lens targeting and the field of views, but I think both the targeting and the field of views that they show are pretty impressive. The first author here has put in a lot of effort and work in getting these to work. I wish I could take some credit for figure 2, but this was actually before my time. But I did help them in later for some of the data in figure 7. Very nice. And then one more question, and again, I don't know if how much you know about this background, but obviously, like, a lot of research has gone into looking at engram cells and kind of the formation of engram cells, and also extinction in the hippocampus. Did the authors at all kind of relate this at all to that work, or just in general in terms of kind of the dynamics of engram formation and extinction, and any comparing or contrasting with principles that they've learned already in the hippocampus?
I can't recall off the top of my head, but definitely, as you pointed out, the group has studied engram cells a lot. This lab has studied engram cells a lot in the hippocampus. I would say that they're probably more in number just because of the anatomy of that structure. And they're probably more involved in the learning phases,
Right.
acquiring the memory. And they've also looked at these engram cells in PFC. Again, off the top of my head, I can't recall what the conclusions were there.
Yeah. Excellent. Yeah. No, I mean, this paper is just another example of also just the powerful techniques that are available now. I mean, the Tonegawa lab obviously is a leader and pioneer in some of these methods, but just the ability to target, not only specific cell types, but activity dependent targeting. I mean, it's really just opening up so many interesting opportunities, and then to be able to combine that with optogenetics and imaging, it's exciting to see this stuff take off and hopefully continue to get applied, to all sorts of different questions and all sorts of different brain networks.
No, and they've really managed to use both the imaging and optogenetics in a complementary manner, and especially the imaging, being able to track the same cells, like in Figure 7, and during different behavior paradigms, is probably what made the conclusion stronger.
Yep. Great. All right. Well, I think we've covered the questions, and this was really great. Thanks to Vardhan for walking us through this interesting publication, and thanks to everyone else for attending and sending in questions. We hope this was scientifically stimulating for everyone. Please stay safe and healthy, and hopefully we can all get back to our regular research activities very soon.
Fingers crossed for that.
Exactly. All right. Well, take care, everyone, and we hope to see you soon again. Bye-bye.
All right. Bye.